Welcome to the Deep Dive, the shortcut to being truly well-informed, where we take complex source material, filter out the noise, and extract the most surprising nuggets of knowledge, just for you.
Today, we are strapping on our molecular goggles, so to speak, and diving into one of the really volatile areas of modern pharmacology, the whole battleground of hyperinflammation.
Yeah.
You've probably heard the term cytokine storm, that, catastrophic cascade of immune activity. It often defines the severity of autoimmune diseases and, frankly, it dictates the worst outcomes in severe infections.
Absolutely. And the challenge for researchers, is huge, is finding ways to actually interrupt that storm.
Right.
So our mission today is, well, pretty specific. We're synthesizing a whole stack of sources to understand the molecular blueprints of four seemingly unrelated drugs.
Yeah, on the surface, they look quite different.
Hydroxychloroquine, Ivermectin, Colchicine, and Famotidine. And we're figuring out how they each manage to modulate the same, central inflammatory machinery.
It's a really fantastic exercise in what you might call translational pharmacology. We're zeroing in on the IL-6 Th17 axis.
Okay.
These are really the core players you need to grasp to understand the scale of that cytokine storm.
Right, so let's define them clearly for everyone listening. What exactly is interleukin-6? Lay that out for us.
Okay, interleukin-6, or IL-6, think of it as a powerful, very widespread pro-inflammatory cytokine, like the master fire alarm for the immune system.
Fire alarm, okay.
When immune cells, say macrophages, detect a serious threat, they just pump out IL-6. It's a rapid systemic signal, basically tells the rest of the body, danger, full-scale defense now.
Makes sense.
The problem, of course, is when that alarm gets stuck on, maximum volume, hyperinflammation.
And if IL-6 is the alarm, then the Th17 axis is you called it the reaction force, the heavier artillery.
Precisely. Th17 cells, that's T helper 17 cells, they're specialized effector immune cells. They don't just appear randomly.
Right.
They're often differentiated from naive T cells by signals like IL-6, often combined with others like TGF-beta. Okay.
And once they're active, Th17 cells produce interleukin-17, IL-17, and that is a major potent driver of chronic severe inflammation. It recruits more inflammatory cells, keeps the whole destructive cycle going.
So the therapeutic goal seems pretty elegant, even if it's difficult in practice. You want to silence that loud upstream IL-6 alarm and then disarm the downstream Th17 artillery that's firing off all that IL-17.
Yeah, I agree.
And the fascinating insight, really, from digging into these sources is just the sheer diversity of strategies these four drugs use to basically accomplish that same task.
It's quite remarkable.
So we're going to break down each distinct key, you could say, that unlocks this anti-inflammatory door.
Right. Let's kick off with hydroxychloroquine, HCQ, and its close relative chloroquine, or CQ. Now, these are drugs many people know for treating malaria or autoimmune conditions like lupus, SLE.
Yeah, they've been around a while. They have. But what's surprising, maybe, is that their main reported action against this kind of inflammation isn't, like, directly blocking a cytokine. It's more about changing the physical environment inside the immune cell.
Okay, so the sources consistently point to this kind of dual action, right? Inhibiting autophagy and also interfering with antigen presentation.
That's the core idea.
And that ultimately rebalances the Th17 response. So let's start with the basics, the chemistry. How does HCQ actually get inside the cell and start, messing with things?
Well, it's a neat trick of its chemistry. HCQ is what we call a weak diprotic base.
Okay, a base.
And that simple chemical property means that within the cell, it tends to accumulate preferentially in compartments that are naturally acidic.
Like lysosomes?
Exactly. Lysosomes and other acidic cytoplasmic vesicles, they just soak it up.
But why do immune cells even have these acidic vesicles like lysosomes? What's their normal job?
Good question. Think of the lysosome as the cell's digestive system and recycling center all rolled into one.
Oh.
It maintains a very acidic pH inside, usually around 4.5 to 5. This lets its enzymes break down all sorts of material-damaged proteins, bits of invading pathogens, old cellular components. It's crucial for quality control and defense.
So if HCQ is a base and it floods into these acidic lysosomes, it's going to neutralize them, right? Raise the pH.
Exactly that. Even a small rise in that lysosomal pH is the crucial physical mechanism we're talking about. It effectively suppresses the lysosome's ability to degrade stuff.
Right.
And that impairs what's called autophagic flux. Autophagy is that internal process of cellular recycling and cleaning. So HCQ essentially jams the cell's garbage disposal.
Okay, that makes perfect sense. A jammed recycling center. Now let's connect that to the immune system. How does impairing autophagy regulate T cells, especially in these hyperinflammatory diseases like SLE you mentioned?
Well, this is where it gets fascinating and maybe a bit counterintuitive. In conditions like SLE, it seems that activated autophagy might actually contribute to the disease state
Really? How so?
by causing an immune imbalance. We see this heightened activity of those pro-inflammatory Th17 cells we talked about, coupled with reduced function of regulatory T cells, or TREGs.
Frags, okay. The peacekeepers.
Precisely. TREGs are vital. They're responsible for putting the brakes on immune responses once the threat is dealt with. So in SLE, you often have too many Th17s and not enough functional TREGs. That's the Th17 TREG imbalance.
So if activated autophagy is contributing to the problem, then HCQ's ability to inhibit autophagy should actually be beneficial for rebalancing the system.
That's exactly what the sources suggest. HCQ treatment and also CQ in related studies effectively helped rebalance these Th17 and TREG responses. This was seen in lupus-like mouse models and also in vitro cell cultures.
Okay, and how does it protect the TREGs? You call them the peacekeepers. What's the aha moment there?
It really comes down to a specific molecule called FOXP3.
FOXP3.
FOXP3 is the master transcription factor. It's basically the instruction manual, the blueprint that a TREG cell needs to function properly as a suppressor. If the cell degrades its FOXP3, the TREG loses its ability to suppress inflammation. It becomes ineffective.
Right.
And our sources confirm that CQ, by preventing that lysosome-dependent degradation, remember, by jamming the garbage disposal, it actually safeguards this crucial FOXP3 protein.
Wow, so HCQ isn't just like trying to put out the fire directly, it's protecting the fire marshal.
That's a great analogy, yes. It preserves the TREG's ability to suppress while also acting on the aggressor cells, the Th17s.
And how does it act on the Th17s directly in terms of cytokine suppression?
Well, its effect on autophagy leads to a significant decrease in the production of that potent inflammatory molecule, IL-17, by the Th17 cells themselves.
Ah, okay.
It also seems to suppress their proliferation, their growth, and their differentiation into mature Th17 cells in the first place.
And there's solid evidence for this.
Yes, the sources detail studies where HCQ, sometimes used alongside prednisone, inhibited both the actual production and even the genetic expression of IL-17 in SLE patients. And in those mouse models, HCQ reduced the levels of IL-17 floating around in the serum, and also IFN gamma, which is another key inflammatory cytokine.
So it's a pretty comprehensive hit on the Th17 side of the axis, downstream.
Definitely a downstream effect,
That's-
primarily.
But let's circle back to IL-6, that main fire alarm we started with. Does HCQ directly shut that off?
Not directly, no, and that's a really important distinction, especially when we compare it to, say, Ivermectin or Famotidine later on.
Right.
HCQ's impact on IL-6 levels seems to be mostly indirect. By disrupting antigen processing and presentation, basically, how immune cells show danger signals to T cells, it sort of dampens the whole environment that causes IL-6 to be produced in the first place. And of course, by suppressing the downstream Th17 pathway, which IL-6 helps activate, it has an indirect effect that way too.
So it's modulating the cellular environment, changing the conditions, rather than, say, directly silencing the IL-6 gene.
Exactly. It's
Yeah.
more about altering the cellular machinery and habitat.
Okay, so if HCQ was more about broad chemical modification, changing the cell's internal pH, jamming the recycling, then ivermectin seems like a completely different approach, more like a scalpel going straight for the electrical wiring.
That's a good way to put it.
We're moving now to its reported mechanism. A really targeted strike against that hyperinflammatory state by inhibiting the STAT3 signaling pathway.
Right. This is a fundamentally different strategy. We're shifting from modifying the T cell environment to directly interrupting the communication cascade inside the cell, that specific signal pathway that churns out IL-6.
STAT3, signal transducer and activator of transcription 3. Sounds technical, but you're saying it's critical. Why is hitting STAT3 such a potentially powerful move against that IL-6 alarm?
Because STAT3 isn't just related to IL-6, it's arguably the central transcriptional command center for a whole suite of inflammatory genes. Think of it like a master dimmer switch for inflammation.
Okay.
When STAT3 gets aberrantly activated, switched on too strongly or for too long, especially in macrophages responding to a threat, it physically goes into the nucleus, binds to the DNA, and tells the cell, make tons of pro-inflammatory cytokines now. and IL-6 is top of that list.
So it directly controls the IL-6 gene.
Our sources confirm that, yes, STAT3 is specifically responsible for activating the transcription of the IL-6 gene itself. So if you want to silence the alarm, you really need to cut the wire connecting the sensor to the speaker. Blocking STAT3 does that.
So how does ivermectin physically cut that wire? Does it just like sit on the STAT3 protein and block it?
This is where we get that real molecular aha moment from the sources. It involves another molecule called PK1.
PK1.
PK1 stands for P21 activated kinase 1. What the study suggests is that ivermectin actually promotes the degradation of PK1.
Degradation, so it causes PK1 to be destroyed.
Essentially, yes. Think of PK1 as a necessary component, maybe like a specific tool or a scaffold that's needed to assemble the machinery that activates STAT3 properly.
Okay, wait. How does PK1 connect to STAT3 then? I'm missing that link.
Right. So PK1 physically interact with, it binds to both Janus kinase 1, JAK1, and STAT3 itself.
Okay.
They form this functional complex, the JAK1 PK1 STAT3 complex. This complex is what actually translocates into the nucleus and effectively switches on the IL-6 gene transcription. It's the fully assembled active machine.
I see. So ivermectin isn't just blocking the signal passively. It's actively promoting the destruction of a key component needed for the signal to be transmitted. It's like dismantling part of the factory assembly line.
Precisely. By compromising STAT3 activities, specifically via promoting the degradation of PK1, ivermectin directly and quite effectively decreases IL-6 production.
Wow.
It's a highly targeted intervention right at the transcriptional source, very upstream of the cytokine storm itself.
And the sources backed this up with some pretty compelling data, didn't they? Showing ivermectin suppressed both IL-6 and also TNF-alpha production.
Yes, significant reductions.
What particularly caught my eye was the mention of modulating infection outcomes by reducing the IL-6-IL-10 ratio. Can you unpack the significance of that ratio for us?
Absolutely. The IL-6-IL-10 ratio is a really critical indicator of how severe inflammation is and how balanced the immune response is.
Okay.
IL-6, as we've said, is strongly pro-inflammatory. It fuels the fire. IL-10, on the other hand, is a key anti-inflammatory cytokine. It's the immune system's calm down signal.
What breaks?
Exactly, the brakes. A healthy immune response needs both. You need the fire to clear the threat, but you need the brakes to stop it from burning down the house. In a cytokine storm, that ratio gets dangerously skewed towards IL-6. Far too much fire, not enough brakes.
So by reducing this IL-6-IL-10 ratio,
Ivermectin helps restore a more appropriate balance between those pro and anti-inflammatory signals. It guides the body away from that runaway hyperinflammation, hopefully towards resolution.
makes sense. And the sources also hinted at some redundancy, that ivermectin might not be just a one-trick pony, potentially influencing NF-kappa B as well.
That's correct. NF-κB is another major pathway kind of running in parallel to STAT3 in many ways, also responsible for driving the production of inflammatory cytokines.
Right.
And studies showed ivermectin also suppressed the activation, the translocation of NF-κB when cells were challenged with LPS, that's lipopolysaccharide, a potent bacterial toxin mimic.
Okay.
So by potentially hitting both the STAT3 and the NF-κB pathways, ivermectin might be essentially putting a double lock on the production lines for key inflammatory cytokines like IL-6.
That seems like an incredibly potent way to stop that IL-6 alarm right at the source.
Yeah.
So if ivermectin is this effective at blocking IL-6 upstream, what's the knock-on effect for our other player, the Th17 heavy artillery?
Well, the effect is indirect, but it follows logically, and it's a powerful one. This is becoming a recurring theme, isn't it? Remember, we said IL-6, often with TGF-beta, is that critical differentiation signal. It tells naive T cells become Th17 cells.
Right.
So if ivermectin effectively starves the system of that essential upstream IL-6 signal by blocking the STAT3 pathway, well, it physically hinders the formation and the proliferation of new Th17 cells.
So you block the signal needed to make them.
And you eventually reduce the downstream IL-17 output because there are fewer Th17 cells being generated. It tackles the source of the Th17 differentiation signal.
Okay, let's shift gears again. Now we're on to colchicine. This is a drug with a really long history, right? Used for gout for ages?
Millennia, actually. It comes from the autumn crocus plant.
Wow. Its mechanism against hyperinflammation, though, seems, well, almost more explosive, because it acts on what you might call the cell's immediate molecular danger sensor.
That's a good description.
The primary strategy reported in our sources involves inactivating the NLRP3 inflammasome.
Right. Now we're diving deep into the innate immune system's really rapid first response mechanisms. The NLRP3 inflammasome isn't just one molecule, it's a multi-protein complex inside the cell.
Okay.
And its main job is to function as a highly sensitive danger sensor. You could call it the cell's internal panic button.
Panic button, I like that. What triggers it?
A really wide range of things that signal cellular stress or damage. Things like certain pathogen components, toxins, crystalline structures like uric acid and gout, even signals indicating mitochondrial damage or potassium efflux from the cell. It's very broadly tuned to danger.
And when that panic button gets pushed, what happens? What's the immediate result?
Activation of the NLRP3 inflammasome pathway is a very rapid, very acute event. And its key function is to process and lead to the release of mature, highly active pro-inflammatory molecules. It's the gateway to immediate, often severe inflammation.
Okay, so it's like an emergency flare gun inside the cell.
Exactly. And colchicine steps in right there at that gateway and effectively shuts it down.
So which specific inflammatory molecules are being stopped by colchicine blocking this inflammasome?
By inactivating NLRP3, colchicine specifically inhibits the processing and subsequent release of two incredibly potent signaling molecules, interleukin-1 beta, IL-1, and interleukin-18, IL-18.
IL-1 beta and IL-18.
Yeah. These cytokines actually exist inside the cell initially as inactive precursors. They're called procytokines.
Okay.
The NLRP3 inflammasome complex contains an enzyme, caspase-1, which is required to literally cleave these procytokines into their mature, active, highly inflammatory forms. Colchicine prevents that whole assembly and activation process.
So it's not blocking a receptor on the outside. It's not inhibiting gene transcription like ivermectin. It's actually stopping the final assembly line for these two critical inflammatory weapons after they've already been made in precursor form.
Precisely. It's intervening at the protein processing level. This is a very effective way to halt that really acute immediate inflammation that's triggered by those danger signals sensed by NLRP3.
Okay, so we've established colchicine stops IL-1 beta and IL-18 production via the inflammasome. How do these molecules connect back to our main target, the Th17 axis? We know the IL-23, IL-17 axis is important there.
Right, this is where the concept of synergy comes back into play. It's about how these different signals work together. IL-1 beta and IL-18 don't typically initiate Th17 differentiation on their own. That's still mainly the job of IL-6 and TGF-beta, as we discussed.
Right.
However, once Th17 cells are present, IL-1 beta and IL-18 act very powerfully in synergy with another cytokine called IL-23 to dramatically promote and enhance the production of IL-17A and IL-17F by those Th17 cells.
Ah, so they're like performance enhancers for the Th17 cells that are already there. They supercharge the output of that really destructive IL-17.
They absolutely do. They accelerate and amplify the Th17 inflammatory output significantly. So therefore, by inhibiting the production of IL-1 beta and IL-18 via the NLRP3 inflammasome, colchicine is interfering with those potent upstream cofactors, the factors that dramatically enhance Th17 activation and their subsequent IL-17 release.
So it's an indirect hit on the Th17 axis again, but
Right.
by removing these key amplifiers.
Exactly. It's a fundamentally essential way to weaken the destructive potential of the Th17 response, even if it doesn't stop Th17 cells from forming initially. It dampens their activity.
Okay, let's quickly recap the strategies so far because they are so different. HCQ modified the cellular pH, jammed autophagy, saved the TREG peacekeepers.
Right, environmental modification.
Ivermectin went straight for the genetics switchboard, STAY3, via PK1 degradation to block the main IL-6 alarm signal.
Transcriptional blockade.
And now colchicine shuts down that immediate danger sensor, the NLRP3 inflammasome, to prevent the assembly and release of IL-1 beta and IL-18, which amplify Th17.
Protein processing inhibition. 3 wildly different molecular strategies, all converging on dampening that IL-6 axis. Okay, our fourth and final strategy brings us to famotidine, a drug most people probably know as, Pepcid or a similar over-the-counter heartburn relief medicine.
Yeah, this one definitely feels like the most surprising entry in our deep dive today. Why would a drug designed to block stomach acid production have systemic anti-inflammatory effects? We need to be really clear here that the sources specifically link its effects on the IL-6 axis back to its established mechanism, right? It's H2R antagonism.
That is absolutely the core linkage proposed in the sources we looked at. And the surprise maybe fades a bit when you realize that histamine H2 receptors, the targets of famotidine, aren't only found on the parietal cells in the stomach lining that make acid.
Ah, okay. Where else are they?
They're actually quite widely expressed across the immune system. You find them on mast cells, which are famous for releasing histamine,
Hmm.
but also on neutrophils, eosinophils, even endothelial cells lining blood vessels. These are all critical cell types involved in inflammatory responses.
Okay, so if histamine binds to these H2 receptors on various immune cells, that binding itself must contribute somehow to the cytokine storm, beyond just allergy symptoms.
It appears so. Histamine itself, acting via H2R, seems to induce a sort of low-level production of IL-6 and another chemokine called IL-8, particularly by endothelial cells.
Okay, low level.
But crucially, this low-level production might act as a kind of systemic primer. So when other, more potent inflammatory stimuli come along, like from an infection or tissue damage, this histamine-induced IL-6 production gets amplified dramatically.
Like adding kindling to the fire before the main logs go on.
Exactly. Therefore, antagonizing these H2 receptors, blocking histamine from binding, effectively mutes this histamine-induced component of inflammation and the subsequent cytokine release it contributes to.
So we're blocking that systemic priming signal. And specifically, which cytokine reduction are we most interested in here, linking back to our IL-6 Th17 axis?
The sources specifically show that famotidine treatment is associated with reduced expression levels of the inflammatory mediator IL-6.
Okay, a direct hit on the fire alarm again.
Yes, and also another mediator called CCL2, which recruits inflammatory cells. But the IL-6 link is key for our discussion. By blocking the H2R, famotidine seems to mute one of the input signals that helps drive the overall production of IL-6 in the bloodstream during inflammation.
Okay, but hold on. If famotidine is normally taken for the stomach, are the doses needed to get this systemic H2R antagonism effect on immune cells safe? Is there a risk of major off-target effects when using it this way in a hyperinflammatory state?
That's a really important practical question. What the sources suggest is that the dosages used in some of these studies, which are often higher than standard heartburn doses, but still within certain clinical ranges, are sufficient to achieve systemic H2R antagonism on these relevant immune and endothelial cells.
Okay.
The idea isn't that it's having some weird off-target effect. It's leveraging its known on-target H2R blocking function, but applying it to the receptors located outside the stomach, specifically on cells contributing to circulating IL-6 levels.
Got it. So it's a known mechanism, just in a different location, the immune cell surface leading to systemic IL-6 reduction.
Precisely. And we should probably briefly mention a potential secondary mechanism that some sources touch on.
Okay.
Famotidine might also potentially activate something called the vagus nerve inflammatory reflex.
The vagus nerve, how does that work?
The vagus nerve is a major nerve connecting the brain to the body, and it has a known role in actively suppressing peripheral inflammation. It's a neural pathway for calming things down. Some evidence suggests famotidine might stimulate this reflex.
Interesting.
It's another potential route to attenuate the cytokine storm, though the direct H2R antagonism leading to IL-6 reduction seems to be the strongest proposed mechanism directly related to our specific IL-6 Th17 axis focus.
Right. So similar to ivermectin then, the primary attack here is on the upstream IL-6 production, but achieved via receptor blockade rather than blocking gene transcription.
Exactly. Different method, similar upstream target.
So what's the powerful indirect consequence for the Th17 axis? Is it the same logic as with ivermectin?
It is essentially the same consequence, yes. Since IL-6 is that crucial factor directing naive T cells to become Th17 cells, the observed reduction in circulating systemic IL-6 concentration mediated by famotidine would automatically and indirectly reduce the fuel for subsequent Th17 differentiation. Less IL-6 signal means fewer Th17 cells develop, leading eventually to less IL-17 production.
So again, by quieting the upstream alarm, IL-6, you prevent the downstream heavy artillery, Th17, IL-17, from being fully formed or activated.
That's the connection.
Wow. Okay, we've just gone through 4 different drugs, 4 seemingly completely different molecular pathways, yet they all seem to converge on dampening this same critical inflammatory axis, the IL-6 Th17 axis. So stepping back, what does this incredible diversity actually tell us about hyperinflammation itself and maybe about how we approach medical intervention?
I think what really stands out is the realization that inflammation isn't controlled by a single on-off switch. It's much more like a complex orchestra with many different sections and signaling pathways playing together.
Yeah.
And these 4 drugs essentially demonstrate 4 quite distinct points of vulnerability within that orchestra, specific instruments or conductors, if you will, that can be targeted for therapeutic benefit.
Right. Let's quickly review those distinct strategies one last time. Just comparing and contrasting them might help solidify the synthesis for, everyone listening.
Good idea. We can almost classify them based on their primary target and the level at which they intervene.
Okay. First up, we had hydroxychloroquine,
Mm-hmm.
HCQ. You called this the cellular environment modifier.
Mm-hmm.
Its strategy was fundamentally changing the internal chemistry, raising that lysosomal pH, which then inhibited autophagy.
Right.
And that disruption then had the knock-on effect of rebalancing T-cell populations, crucially protecting the TREGs and suppressing the Th17s, so addressing downstream consequences by altering the cellular habitat.
Exactly. Next, we had ivermectin. This one was the genetic transcription blocker.
Right, the scalpel.
It went straight into the command center, promoting the degradation of PAC1, which effectively shuts down that critical STAT3 transcription pathway.
The pathway mainly responsible for mass IL-6 gene activation.
Correct. So that's a very precise upstream intervention, right at the level of gene expression.
Okay. Then third, colchicine. This was the innate immune emergency brake.
Yeah, hitting the inflammasome.
It shut down that immediate danger sensor, the NLRP3 inflammasome, stopping the final processing and activation of key pro-inflammatory cytokines like IL-1 and IL-18, which act as Th17 enhancers.
So intervening after the gene is transcribed, but before the protein becomes fully active at the protein processing level, and targeting those amplifiers.
Got it. And finally, famotidine. This is the targeted receptor blockade.
Mm-hmm.
By acting as an H2 receptor antagonist, specifically on immune and endothelial cells, it reduced histamine's contribution to cytokine release, leading directly to a systemic reduction in IL-6 levels.
So a clean block of one specific input signal, histamine signaling via H2R, that contributes to the overall inflammatory milieu and IL-6 production. Another upstream intervention, but via receptor antagonism.
The relevance, then, for you listening is perhaps recognizing that complexity in the body doesn't always require an equally complex drug. Sometimes it requires a drug with a very specific, highly targeted, and leverageable mechanism.
Absolutely.
It really underscores that multiple, incredibly distinct molecular targets, from messing with pH, to degrading a kinase, to blocking an inflammasome, to antagonizing a receptor, can all potentially lead towards the same clinical goal: mitigating hyperinflammation by calming this IL-6 Th17 axis.
It also tells us something vital about the sophistication, maybe the redundancy, built into the inflammatory response itself.
How so?
Well, the body has so many overlapping systems involved: STAT3, NFT, NLRP3, various cytokine receptors like H2R contributing, that hitting just one key pathway, as each of these drugs reportedly does, seems to provide a window to dampen the overall response significantly, hopefully without completely collapsing the entire immune system, which you obviously need.
Yeah, that's a crucial balance. It's almost staggering, though, that we use these 4 drugs, some very commonly, yet their molecular strategies are, as you said, like comparing a wrench, HCQ's pH effect, to a laser pointer, ivermectin's STAT3 targeting, to maybe a fire extinguisher component, colchicine's inflammasome block.
It's a testament to the different ways we can interact with complex biological systems. Hashtag outro. Final thought.
Well, this has been a deeply satisfying exploration, I have to say, really getting into the weeds of molecular pharmacology and how these drugs modulate the immune system. Thank you for, bringing these sources together and letting us connect these dots for you. Absolutely. And as we wrap up this deep dive, we want to leave you, our listener, with a final, maybe provocative thought, something that builds directly on the synthesis we just completed, looking at these quite distinct strategies.
We sort of established, didn't we, that these 4 drugs, at least in terms of how they impact the IL-6 Th17 axis, naturally seem to fall into 2 broad functional categories based on where they primarily intervene.
Right. On one side, you've got what we could call the upstream blockers. That would be like ivermectin and famotidine.
Mm-hmm.
Their main strategy seems to be targeting IL-6 production itself. They're trying to cut that initiation signal, essentially blocking the spark that gets the big inflammatory process going.
Exactly. Then on the other side, you have maybe the downstream modulators, thinking here of HCQ and colchicine. Their primary impact seems to be more on modulating the T-cell differentiation, or the enhancement of T-cell function, like protecting FOXP3 and TREGs, or blocking the IL-1 amplification of Th17s. They seem to be suppressing the resulting cellular proliferation and action, or maybe dampening the fuel once the fire has started.
Okay, spark versus fuel, upstream versus downstream.
Yeah.
So here's the thought for you to ponder. When you're faced with treating an acute, rapidly escalating hyperinflammatory state, like a cytokine storm.
Yeah.
What does synthesizing the source material suggest might be the optimal approach?
Is it strategically better, perhaps, to prioritize blocking those initiation signals upstream the IL-6 spark with drugs like Ivermectin or Famotidine, trying to prevent that Th17 cascade from ever really getting established?
Or is the more robust approach actually to target the downstream effects, to use drugs like HCQ or Colchicine to dampen the resulting cellular proliferation and action, maybe assuming that by the time you intervene clinically, that initial IL-6 signal might have already happened, and now you need to control the fire itself?
It's that critical clinical strategy question. Preventative signal blockade versus rapid effector suppression. Block the spark or control the fuel. It's something for you to mull over, connecting these fascinating molecular mechanisms we discussed to the real-world challenge of treating hyperinflammation.
Until the next deep dive, keep learning.